Ever wonder why we refer to some parts of our DNA as junk? For an extended period, a significant portion of our DNA, known as non-coding DNA, was misunderstood and dismissed as superfluous. However, a shift in scientific understanding has emerged, revealing the far-reaching implications of this seemingly useless genetic material. No longer considered junk, non-coding DNA is now recognized as having a pivotal role in our biology. Today, we're diving into the world of non-coding DNA. Did you know that 99% of our DNA is non-coding? It's a fascinating fact, isn't it? This part of our genetic blueprint, known as non-coding DNA, doesn't directly code for proteins, yet it's far from being useless. Contrary to what its name might suggest, non-coding DNA isn't just a passive bystander in the theater of our genetics. It's an active participant, a behind-the-scenes maestro, if you will. Its roles are many and varied. Non-coding DNA is instrumental in regulating gene expression, influencing how, when, and where genes are turned on or off. It's also crucial in shaping the structure of our chromosomes, helping to keep our genetic material stable and intact. Furthermore, it's responsible for producing non-protein coding RNAs, which are vital players in our cellular machinery. So it's clear that non-coding DNA holds the keys to more than we initially thought. It's not just about the proteins, but about the intricate dance of life happening in every cell, every moment. Imagine having the power to switch genes on and off. That's exactly the role of regulatory elements nestled within the vast expanse of our non-coding DNA. Think of these regulatory elements as genetic switches, gatekeepers that control the intricate process of gene expression. They determine when a gene should be activated or silenced, effectively orchestrating the symphony of biological processes that define our existence. But how do these genetic switches work? They interact with proteins called transcription factors. These transcription factors bind to the regulatory elements, acting as the key to turn the genetic switch on or off. Regulatory elements have diverse forms and functions. Some are located close to the gene they control, while others might be positioned quite a distance away. Despite their varying locations, all of them play a crucial role in managing the flow of genetic information, ensuring that the right genes are expressed at the right time, in the right cells. The interplay of regulatory elements and transcription factors forms a complex network of control, a genetic dance that influences everything from our physical traits to our susceptibility to diseases. As we delve deeper into the world of non-coding DNA, we're beginning to appreciate the profound impact these genetic switches have on our biology. These genetic switches are the conductors of our genetic symphony. Just like traffic signals, non-coding DNA directs the flow of genetic information. It's an intricate network, a sophisticated system of control that governs our genes. Within this network lie regulatory elements, the unsung heroes of our genetic operations. Let's take a closer look. First up, we have promoters. Located near the start of genes, these elements serve as docking sites for the machinery that reads and transcribes DNA. They're like the on-ramps to a highway, guiding the flow of genetic instructions. Next, we encounter enhancers and silencers. These elements might be located far from the genes they regulate, but distance doesn't weaken their influence. Enhancers are like the accelerators, boosting gene activity when needed. Silencers, on the other hand, are the brakes, shutting down gene activity when it's no longer required. Then there are insulators. These elements act as buffers, preventing enhancers and silencers from accidentally turning on or off the wrong genes. They're the traffic barriers of our genome, ensuring each gene follows its prescribed path. And finally, we have locus control regions, or LCRs. These elements oversee clusters of related genes, coordinating their activity like a traffic control center. Each of these regulatory elements plays a unique yet interconnected role in the grand symphony of our genes. They ensure that the right genes are expressed at the right time, in the right cells. Non-coding DNA is not just a bystander but an active participant in our genetic operations. Non-coding DNA also encodes various RNA molecules that play critical roles. Indeed, the story of non-coding DNA doesn't end with the orchestration of protein synthesis. Let's delve into the world of non-coding RNAs, the silent workhorses of our genetic machinery. In the bustling city of our cells, non-coding RNAs are like dedicated city workers, carrying out essential tasks mostly unseen. For instance, transfer RNAs or tRNAs serve as the link between the genetic code and the protein-building amino acids. They are the interpreters, making sure that the right amino acid is picked up for every codon in the messenger RNA, ensuring the correct translation of the genetic code into proteins. Then we have microRNAs or miRNAs, the tiny yet powerful regulators of gene expression. They bind to messenger RNAs and prevent their translation into proteins, effectively silencing genes when needed. In this way, they play a crucial role in cellular functions, development, and disease prevention. Long non-coding RNAs or LNCRNAs are another fascinating group. They're like the multitaskers of the cellular world, involved in gene regulation, chromosome modification, and even acting as molecular decoys. Each LNCRNA holds a unique role, and their functions are as diverse as the number of LNCRNAs themselves. So you see, non-coding DNA gives rise to a plethora of non-coding RNAs, each with a unique, crucial role in our genetic orchestra. These RNAs are the unsung heroes of our genetic machinery. They may not grab the headlines like proteins, but without them, life's symphony would be out of tune. Our genetic integrity owes much to non-coding DNA. This silent hero of our genome plays an indispensable role in maintaining the structure of our chromosomes and safeguarding our genetic information. Let's take a journey down the microscopic corridors of our cells, where chromosomes, the carriers of our genes, reside. These chromosomes are not just haphazardly strung together, they have a specific structure. And that's where non-coding DNA comes into play. It provides the scaffolding for the chromosomes, helping them maintain their shape and stability. Some parts of this non-coding DNA form telomeres, the protective caps at the ends of chromosomes. Like the plastic tips on shoelaces, telomeres prevent chromosomes from fraying and sticking to each other, which can lead to genetic disorders. Then we have satellite DNA, repetitive sequences that make up a significant portion of our genome. While their function remains largely enigmatic, they are thought to play a role in organizing the chromosome within the cell nucleus. Finally, there's heterochromatin, a tightly packed form of DNA that helps keep our genes in their correct locations on the chromosomes. It's like a genetic anchor, ensuring that our genes don't float off into cellular space. So you see, non-coding DNA is not just a bystander in our genome, it's a key player in maintaining the integrity of our genetic information, ensuring that the story of our genes is told accurately and efficiently. So, non-coding DNA is also the guardian of our genetic information. Introns and intergenic regions, parts of non-coding DNA, are still a mystery. Introns, the sequences excised from mRNA during protein synthesis, are not just discarded genetic material. They're hiding places for regulatory elements like enhancers and silencers subtly controlling gene expression. Scientists are investigating these regions, delving into the roles they play in the dance of DNA. Intergenic regions, on the other hand, are the stretches of DNA sequences between genes. Once thought to be mere genetic deserts, they're now considered potential gold mines of undiscovered regulatory elements. Their vast expanses could hold the keys to further unlocking the mysteries of gene regulation. Every intron and intergenic region is a piece of the puzzle, a part of the grand tapestry that is our genome. As we continue to unravel, to decode, we edge closer to fully understanding the power and potential of non-coding DNA. These regions could be the next frontier in our understanding of non-coding DNA. Scientists are on an exciting quest to decode the non-coding DNA landscape. This expedition into the genetic unknown is revealing new layers of complexity within our genome. Each day, researchers are identifying novel regulatory elements and structural components that were previously hidden within our DNA. The exploration is far from over. With every step, there's a deeper appreciation of the intricate dance of life, showing us how these non-coding sequences influence gene regulation, development, and even disease. From complex diseases like cancer to developmental disorders, and even our unique human traits, non-coding DNA plays a pivotal role. The mysteries of non-coding DNA are slowly but surely being unraveled. With advanced technologies and innovative research approaches, scientists are illuminating dark corners of our genome. As we continue this journey, we're not just decoding DNA, we're deciphering the very language of life itself. Every day we're uncovering more about the profound impact of non-coding DNA. From junk to a master regulator, non-coding DNA has come a long way. Its story illustrates how science evolves, transforming the dismissed into the indispensable. This unsung hero of our genome regulates genes, influences cellular functions, and safeguards our health. It's a vast, uncharted landscape teeming with potential and promise. As we continue to decode the language of DNA, we're gaining deeper insights into the intricate mechanisms of life.